And bonjour. And actually, this time I can really say bonjour, Kalpana, because I'm in France. I
Yes.
will be in France for a few more days, then back to Chicago for AECR. And you're listening to the Building Public podcast from the LARVOL CLIN team at LARVOL. We are building the oncology superintelligence ultimately, and CLIN is a stepping stone in that endeavor. CLIN is a highly curated Wikipedia of clinical trial data in cancer. We are here today with Kalpana to talk about a very important feature of CLIN, and a feature that we've developed over the last 12 months or so that many of our customers like, and it's a landscape that ultimately is probably the most advanced element on the way for a pharmaceutical company to build a target product profile, a TPP. So we're going to introduce you in more details to that feature. Before that, Kalpa, let me put you on the spot. How would you introduce LARVOL CLIN to someone who doesn't know too much about it in a couple of minutes? And then we'll transition to our landscape feature itself.
Yeah, sure, Bruno. Uh, people, like most people know about CLIN for now, but if those who don't know about CLIN, it is something like they might be knowing about Wikipedia. So I would define CLIN as a Wikipedia for oncology trials. We have all the data related to an oncology trial, right from the trial details, sponsor, whether it is a randomized trial, registration, if the FDA information, the data, adverse events, KM plots, forest plot, and advancing into AI, we have the pseudo IPDs, NMAs, everything, whatnot. Everything is here. So,
And the reason why pharmaceutical companies, and actually also some academic groups, increasingly a lot of academic groups, know about us, use us. We have a free version for the academic group. Is that the the data we have in many cases is very hard to get, and very hard to get in a single place, especially the outcome data. So the
correct.
slides, the posters, the all the manuscripts, and the color brought by a panel of 6,000 oncologists. So we track 6,000 oncologists on X, and each time they mention something about a cancer clinical trial, our AI platform extract the sentiment and the details of their post, and we put it all together. Essentially, when you look at CLIN, you're not looking at Kalpana and Bruno's opinions about this clinical trial, though we have some opinions that sometimes we share, but really you're looking at the distillated picture of 6,000 oncologists. And out of these oncologists, we actually rank them very carefully, including through a survey based on the proeminence of their voice. So out of the 6,000, we have 100 of them across all of the major specialties that are considered top voice, and their opinion is highlighted more prominently. So now that
Yes.
we created the setup for CLIN, let's talk to pipeline. Tell us, I'm sorry, let's talk to the landscape feature.
Yes.
So let's go through that feature with you.
Sure. Okay. We have landscapes, some people call them as data tables. And we show the landscapes mainly in three places currently. One is at the trial 360 page. We call this as the trial 360 page because this has every detail of one trial. So under the landscapes tab, you see all the landscapes or data tables that are related to this particular trial, and these are publicly available. So we call them as third party landscapes, which are built by someone else, and we are consolidating and keeping them at one place. And the second place is in the main landscapes tab, where we show proprietary landscapes which we are building specifically for any customer. I'll dive in a little later about the proprietary landscapes. And there is one more feature which we have already discussed in the previous podcast regarding the key population. Under the key population, we are showing n number of features. like you can filter with a specific tumor type, and under that we see the key population definition, something like first line metastatic NSCLC. If I click on it, so under this you see the list of all the landscapes or data tables. This again are the third party landscapes. So given the brief introduction about this, for example, this is one landscape and this is one landscape. We source it from different places. It can be from Twitter, it can be from meta-analysis study, it can be from pipelines. And so now
A comment about these third-party landscape. I mean,
yeah,
obviously, the most recent landscape in a particular population is going to be usually the most authoritative. So
correct.
the landscape that was shared in the last Congress is typically the most important one for that key population. And what I found super helpful, and we heard that from our customers as well, is that a good way to know what trials are related to another trial is that they are co-mentioned, they are compared in
Yeah,
a given landscape. So that feature allows us, if we look at again one population, to know all of the top trials
thanks.
ranked by proeminence for a given for a given not just tumor type, but again a specific setting and staging. So if you're interested in EGFR mutated first line non-small cell, we have a very objective way to know the top 20 trials that people are talking about, because these are the trials that are mentioned together either in a landscape published by reputable sources, or sometimes in meta-analysis. So I think that these are very useful rankings of relevant trials.
Yes, that is very well said. Yeah. Okay. Uh, just that you mentioned, like the most relevant landscape is from the previous conference or the most recent conference. Okay. Sometimes what happens is like there might be this kind of data tables presented in ESMO 2024. But if there is any recent data available, for example, for Mariposa trial, there is a recent data that is available from ELCC. There might not be any publicly available landscape or data table. So how are we going to address it? So that is the rebuild feature. So would you like to give a color on it?
Yeah, sure. So what we decided to do is that in each of these key populations, we select a third-party landscape as the most relevant, most up-to-date one, and then we rebuild it. So we extract the data. We of course got to refer and quote and credit the party that issued that initial landscape, and we rebuild it and update it. So we adapt it essentially. We move it to an electronic format. You got to see maybe an example here, and then as soon as there is new data to update that that template, we do it, and we update on our own that landscape. So, for example, the yellow cell that you can see on that screen is new data that wasn't available, or maybe an immature version of that data was available, and now new data has been issued. One thing we could do, this is built in public, so we have ideas in public, by the way, and feel free to comment or ask us questions as you're listening. But one thing we could do, perhaps, Kalpana, is that the source for what's in yellow could
Hmm.
itself have a highlight in yellow, so that it's
Yeah,
super clear. Maybe it's already the case. No, not yet, maybe.
not
So
it.
it's super clear what it means, and it's
Hmm.
super clear what the eyes is drawn essentially to what is new. Is that in that landscape? We updated
Correct.
the landscape, we rebuilt it, we refreshed it. We keep, of course, crediting the original source. It's an
Yes.
adapted, adjusted landscape, and now it become a living and breathing survey of that tumor setting. Now, one
Yeah.
other feature we talked about that we're considering is to have two versions of that landscape. One version is a compact one, the one that you
Yes,
can post on social media or put on a on a PowerPoint if you're
presentation.
presenting to executives in a pharma company. But obviously, it's not all the data. The whole data is probably 10 times this. You have all the subgroups, you have maybe some endpoints that only exist in a couple of these trials, and all of these are not selected to be shown. So if we look at really all of the available data for these, let's say 5 trials,
Mm,
you've
mm.
got 10 times more the data. So we have also an extended version of these landscapes. So some people are interested in the compact version, the briefing version, and the compact version, and others the more expanded one. And so we want to add an expand button for more advanced use, where users can really look at the entirety of the available data with
Yes.
comparison, of course, but also with a lot of gaps, because not every trial would have comparable data points.
Yes, that's very true, Bruno. And that is why building this landscapes, be it be the customized landscapes or a rebuilt landscape, adding more color to the existing landscape which are previously posted will be more value. And it is not like an easy work to do. So we are there to help.
And so for the audience, Kalpana and her team are working so hard in not just building the interface, the front end of our platform, but also obviously in curating the data in the back end. And although AI is increasingly helping them uh, so that they can sleep more, there's still a very important step of QCing the AI and verifying that the data is correct.
Yes. Okay. Adding more color to the landscapes, like this data table might be available publicly, but our touch is always important. So we have the KM plots and forest plots related to whatever the trials that are discussed. So you can see the trials that are discussed in the data summary. We have the KM plots plotted over here. We have the forest plot, and we even have the Twitter reactions, which are arranged based on the timeline. I'm sorry, it is loading. Yeah. So you can see like Flora has 40 tweets, Flora 2 has 355, Mariposa has 514, Papillon has 138 tweets. So the amount of tweets also shows like the importance of one trial. Recently, the Mariposa has drawn more attraction. That is why you see more number of tweets over here. And also you can see the segregation based on the sentiment, like how many negative, how many not rated, mixed, positive, practice informing, practice changing. That data also is displayed pictorically over here. So by one glance, you'll be able to understand, okay, so which is the most important trial among this. By the data seeing, I would say like Mariposa. The second important might be a Papillon trial or a Flora 2 trial.
That you
And
can, of course, click on any of these bars, and you can directly access the underlying commentary tweets. And
yes,
again, this is AI generated, but we do provide an element of QC, in particular of the negative comments, which of course often are the most interesting one. And for the negative comment, we want to make sure in particular that these have been QC'd, that the AI did a good job identifying not only that they're negative, but also why they're negative, because we're indicating whether or not it's due to the commentary about toxicity, or in some cases about cost, or in some cases about trial design. Okay.
yes. Sometimes it's a mixture also, like you see over here. It might be toxicity and financial reimbursement. Sometimes it is only toxicity. So,
Now, one of the future direction
mm,
of these might actually be to make an NMA, a network meta-analysis, in addition to a
yeah,
simple meta-analysis available. So we're looking at using AI and more standard biostatistical tools to provide a productized picture, a scalable picture of these, including, yes, including the timeline of these.
Yeah. So just to give a picture over here, like under the timelines, we'll be able to see like for Flora 2, there are a number of milestones. So we display all of them. Like obviously they are hidden for the first glance, but upon expanding, you should be able to see all the milestones. When is it
Mm.
approved? What is the FDA action date? What is the primary completion date? You have all the data over here. So I would say this is the LARVOL touch. Though this
Yeah,
data is taken publicly. Yeah.
the LARVOL touch. I like it. So we are at the end of our 15 minutes together for this Building Public LARVOL CLIN podcast with Kalpana. Thank you for joining us again, Kalpana, on
My
this
pleasure.
podcast. And looking forward to seeing you another time. For the audience, please join us. Add to your calendar to join us next week with a new episode. And in the meantime, like, repost, subscribe to our channel. Thank you very much. For those of you who will be at ACR, I hope to see you there. Please say hello. I'll be there with my purple cap, so you can easily recognize me in the crowd. And obviously, for those who don't go to ACR but will be at ASCO, we will be at ASCO. We probably will have a special issue of either this podcast or the sister podcast, LARVOL Oncodata, regarding ESMO breast as well, by the way. So keep your eyes out for that as well.